everyone, welcome back to another important session from your favorite class Brain Booster. I'm your Najishwan, and in today's topic we will be learning about a very interesting topic of lac-operon. I assure you beta, you will never learn this topic more easily like this. But before that, you need to stick to the video till the end. So let's get started. First, start with the name. Here lac-operon consists of two terms. First is lac, which is lactose. Lactose is nothing but a milk sugar. Lactose is nothing but a milk sugar. And operon simply means a single switch that lights up multiple bulbs. Single switch that lights up multiple bulbs. Alright? Now what does it mean? This single switch is referred to a common promoter. Common promoter. This common promoter, it expresses multiple genes. Okay? Now there are two scientists that are Jacob and Monod. Now two scientists named Jacob and Monod, who discovered this lac-operon. If we talk about the structural organization of lac-operon, so here we have a simple mnemonics through which we can remember the structural organization of a lac-operon. That is PPO-ZYA. Seems funny, right? So what this PPO-ZYA means? Let me decode this PPO-ZYA. Let me decode each and every alphabet of this PPO-ZYA. Now this P stands for promoter. Alright? P stands for promoter. I for inhibitor. Or you can say repressor. A promoter, what is a promoter? A promoter is a sequence where DNA polymerase binds. Alright? O stands for operator. What is the role of this operator sequence? Operator sequence is such a DNA sequence where this repressor protein binds. Now, if I talk about this Z, Y and A, this Z, Y and A in lac-operon, these are structural genes that codes for various enzymes. Z codes for an enzyme that is beta-galactosidase. P codes for permease and A codes for trans-acetylase. Together, this Z, Y and A are structural genes of the lac-operon. These are the structural genes of the lac-operon. Beta-galactosidase, permease, and trans-acetylase. Together these are termed as structural genes. Structural genes. Okay. Now we can understand this lac operon, or we can remember this lac operon through another mnemonics, that is ARSO. That is ARSO. What this ARSO means? Here R means regulator, S stands for structural genes. Structural genes. O for operator, and P stands for our promoter sequence. Alright, so either PPO-ZYA or ARSO. You can remember the structural organization of lac operon through these two mnemonics. Now, when lactose is present, lactose is present where? Lactose is present in culture media. See, this lac operon is only seen in prokaryotic system. It's only seen in prokaryotic system where multiple genes express under a common or single promoter, because there is a polycystonic mode of gene transcription. Alright, so when a bacterial cell goes inside a media and lactose is present, what does this lactose do? This lactose enters into the cell through permease. Permease will help to enter the lactose inside the cell, alright? And beta galactosidase helps to break down the lactose into glucose and galactose. So basically, this lac operon switched on when lactose is present inside the medium. That's why lac operon is an inducible operon. Lac operon is an inducible operon. Alright, beta? Okay. Let's move to the next point. Now coming to the NCERT part. That when lactose is present, in that situation, what will happen? We have this PPO-ZYA, right? We have this PPO-Z, Y and H. When lactose is present, what will happen? The repressor mRNA will be transcribed. From that, a repressor molecule will bound. Alright, from that, a repressor molecule will be formed. From the repressor DNA, the repressor mRNA will be transcribed and it will be translated into a repressor protein. But due to the presence of the inducer, that is our lactose, it will repress the repressor. It will repress the repressor. What does it mean? Now, this inhibited repressor is unable to bind to the operator. Right? This repressor inhibitor is unable to bind to the operator. As operator sequence is not occupied, so promoter is free for the binding of DNA polymerase. As operator is not occupied, so RNA polymerase will be able to bind to this promoter sequence. And what will it do? It will transcribe all the structural genes. From the structural genes, beta galactosidase, permease, and trans-acetylase. Beta galactosidase, permease, and trans-acetylase will form, which will break down the lactose into glucose and galactose. I hope you enjoyed the session. We will continue in the next session. Thank you.